Hello, I want to go over just some quick teacher notes about the Enzyme lab investigation that would hopefully make this lab run a bit more smoothly with your students. So starting with the catalase, the source of the catalase. There are many sources of catalase that you could use for this lab because it's found in living tissue. I've personally found what's mentioned in the lab, the red potatoes, to be most effective as far as getting the right amount of oxygen foam. I've actually done this, I believe, with russet potatoes. Do not get as much foam with that. What you want to be able to do is use 2 or 3 kind of regular size red potatoes, and then when you prepare them, I would take the skin off and cut them into little bits, almost like cubes, on a cutting board, and then put it into a blender. You can use a juicer. That's mentioned in on the Google Doc teacher notes that a juicer is an option, but most teachers and myself tend to use a blender. And you put them in a blender with about 200 milliliters of water. One thing to note, if you're noticing that you're not getting as much oxygen gas as you would like in order to measure, you could lower the amount of water and maybe increase the amount of potatoes just to increase that catalase concentration. After you blend it in so that it's kind of like a frappe, you want to either strain it through cheesecloth into a large beaker of some sort, or what I've been using is just a typical wire mesh strainer, and that works really well because you don't have to keep buying cheesecloth every single year. And then for the actual lab setup, everybody is going to need hydrogen peroxide. Something to be aware of, hydrogen peroxide has a pretty limited shelf life. So I usually try to get fresh hydrogen peroxide each year. If you have some that's on your chemical shelf, just kind of take a look at the expiration date to make sure that it's not expired, because that would definitely affect your results. And then I use small plastic pipettes for the students to pipette in how much they need into 10 milliliter either plastic or glass cylinders. And like I said, they all need hydrogen peroxide, they all need distilled water, they all need catalase as their enzyme. So what I've done, and you can do this different ways, a lot of teachers will set up stations for the chemicals just to make it more efficient for the students. But what I've done is I've taken the hydrogen peroxide, the distilled water, and then the catalase, and going back to the catalase, that does need to be fresh. So if you have 3 biology classes spread out throughout the day, I will make fresh catalase for each class, because you will notice that the catalase tends to degrade as the day goes on. But this way, when the students come to start their lab, if you have all 3 chemicals ready to go into a little dish, a plastic dish like that, then they can just grab what they need and take it to whatever lab station where they're going to be working. I even include the ruler. So each group will have a ruler, and I also include a graduated cylinder, which would need to be rinsed out to prevent contamination, or you can throw 2 or 3. I have limited space, so you can put that in there. So they've got their ruler, their graduated cylinder, and then the 3 small, these are actually 100 milliliter Pyrex glass beakers that fit pretty easily in there. And then they take what they need to their individual lab station. As far as the different variables go, each variable tends to have the same setup with a few exceptions. With the temperature variable, you're going to need some water baths to change the temperature. I've tried it before with hot plates. Those are very time consuming, trying to get those target temperatures that we're looking for, the 30, 40, and 50 degrees. These I purchased off of Amazon, super cheap, and they work great. You actually set the temperature that you want, and in about 5 minutes or less, it's that temperature. For the room temperature, obviously you don't need to worry about that. You would just need a thermometer to just double check, because room temperatures do vary from classroom to classroom. For the cold temperature, you want to try to get as close to 10 degrees as possible. And what I've done is just given that temperature group a 250 or 400 milliliter glass beaker of ice water, and they check that ice water temperature. If they're trying to get it colder, they can add more ice. If they can pull ice out to try to get the temperature up, depending on where they're at. So the temperature group will be needing the water baths. So I would put the water baths as close to the temperature group as possible. One thing I've noticed, if you have different levels as far as your students go, if you have a really high functioning lab group, give them the temperature variable. That takes the most time and it's a little bit more complex than some of the other variables, as well as the pH. If you're running pH, give a group that is maybe a little bit higher functioning as far as that goes. It will take longer. It's a little bit more of a challenge, a little bit more work because you have more to do compared to the enzyme and substrate concentration. Make sure if you are working in the pH group with hydrochloric acid or sodium hydroxide, make sure each student has access to goggles that they're going to wear. It's super important from a safety standpoint. And the last thing I would mention as far as the test tubes go, whatever test tube rack you're using, you do want to have a test tube rack that has 2 rows of openings because you've got a row of large test tubes and a row of small test tubes. So this is one of the test tube racks that I use in my classroom, and I actually label what their variable is because each group is going to focus on a different variable. And then I print out the instructions for the variables separately on colored paper. This is actually cardstock, and it's basically set up so that it's like a checklist. So you can have your students each take a different role. One person could be in charge of the directions, the protocol, making sure things are getting done correctly. And I actually just put the instructions right there with the test tube rack, and I tell the students, come on up, grab a test tube rack. The color of the instructions corresponds to whatever variable that group's going to be focusing on. So hopefully that's helpful.